Welcome to your 3-minute crash course about the cell cycle or mitosis. Every second, millions of cells in your body are dividing. Skin cells replacing old ones, blood cells being renewed. Even as you watch this video, your cells are following a precise, regulated process called the cell cycle. So let's break it down into some simple steps. The cell cycle is an ordered sequence of events that a cell goes through as it grows, copies its DNA, and divides into 2 new cells. The 2 major parts are interphase, where the cell grows and prepares, and cell division, where the cell actually splits. Most of a cell's life, about 90%, is spent in interphase. The first step of interphase is G1, where the cell grows, produces proteins, and carries out normal functions. This is also where the cell checks, do I have enough nutrients, is my environment safe? And some cells, like neurons, exit here and enter G0, a non-dividing state. The next step in interphase is S phase, where DNA is replicated. Every chromosome is copied, forming 2 sister chromatids. This step is critical because errors here can lead to mutation. In G2, the next step, the cell grows more and checks, was my DNA copied correctly? Only if everything looks good does the cell move on to division, or the M phase. The next stage is mitosis, which is the division of the nucleus, and it happens in 4 stages remembered as PMAT, or prophase, metaphase, anaphase, and telophase. In prophase, the chromosomes condense and become visible. In metaphase, the chromosomes line up in the middle of the cell called the metaphase plate. This alignment ensures each cell gets a new DNA. In anaphase, the chromosomes separate and are pulled to opposite sides, and in telophase, new nuclear membranes form and the chromosomes decondense. The next stage is cytokinesis, which divides the cytoplasm. In animal cells, a cleavage furrow pinches the cell in 2. In plant cells, a cell plate forms because of the rigid cell wall. The result is 2 genetically identical daughter cells. The cell cycle is tightly regulated by checkpoints controlled by proteins called cyclins and CDKs. These checkpoints prevent DNA damage, incomplete replication, and uncontrolled division. When regulation fails, cells divide uncontrollably. This is cancer. Understanding the cell cycle helps scientists discover cancer treatments and understand growth, healing, and development. In summary, the cell cycle is a carefully regulated process by which cells grow, copy their DNA, and divide to form 2 identical cells, asexually. Thank you for watching this 3-minute BioRun, and I hope this helped.